Very good morning students, this is your Prasanna ma'am from NEET World. Now we are going to discuss about the most important and crucial chapter, that is Principles of Inheritance and Variation, which is important both for NEET and EAMCET. From that, now we are going to focus on one gene inheritance and deviations of Mendelian laws or principles from monohybrid cross. So before entering into the chapter, it is very much important to know or to understand about all these terms so that we can apply this chapter in a better way. The very first term is inheritance. What is inheritance beta? Transfer of characters from parents to offspring, or transfer of characters from one generation to the other generation is called as inheritance. What is variation? Variation is nothing but the difference or the degree by which progeny is varying from the parent. The degree by which progeny or offspring is differing or varying from the parent is said to be variation. What is a character? Character is nothing but any feature of an organism, be it skin color, height, or eye color, all these are characters. Trait is the distinguishable feature of a character. Let it be, for example, height is a character, trait is either tall or dwarf. Or else you can also define trait as the character which is transferred from parents to offsprings, or the character which is inherited is also called as trait. Trait is also called, what you can call as trait? Character which is inherited. Character which is inherited is also called as trait. Now when we need to discuss about factor, gene, alleles, homozygous, heterozygous, let us understand this by taking a human cell. Human cell, if we take a diploid human cell beta, diploid human cell contains how many chromosomes ma? 46 chromosomes. Diploid human cell contains 46 chromosomes, of which these chromosomes are present in the nucleus of a human cell, right? So, of 46 chromosomes or 23 pairs, how many are allosomal and how many are autosomal? 22 pairs are autosomal and one pair is allosomal, which undergoes, which is into the process of sexual reproduction and transfers the traits or characters into a progeny. So let us assume there are two chromosomes, or half set of this one pair, half set of chromosomes or haploid pair of the chromosomes, both for male and female. Haploid set of chromosomes both for male and female. Just a second. Yeah. So when it is coding for multiple characters, let us assume only one character, right? Let it be height only. Height. Let parent is homozygous tall and male parent is homozygous tall, female parent is homozygous dwarf. So yes, these two are said to be homologous chromosomes, as they are clearly coding for a contrasting traits, and they are located at the same loci. Two alleles located at the same loci of two different chromosomes, then these two chromosomes are said to be homologous chromosomes. So now what is a gene? Gene is nothing but, gene codes for a specific character or a trait with clear contrasting pair, which predicts a clear contrasting pair of a character. Factor is nothing but the name given by Mendel. Now we are calling it as gene. Gene we can call chemically as functional unit of inheritance or a segment of DNA. Gene we can call it as what ma? Functional unit of inheritance or segment of DNA. Factor is the name given by Mendel. So when these two chromosomes undergo fertilization and involved in the zygote formation, let us assume this pair of chromosomes are crossed and the zygote is heterozygous The zygote is heterozygous tall. Zygote is heterozygous tall. So yes, here it is clearly showing a contrasting pair of characters, but always do remember dominant allele is masking the effect of recessive allele. So it is heterozygous tall. Dominant allele always masks the effect of recessive allele. So the zygote is heterozygous tall, and phenotypically it shows tallness only, tallness character only. So yes, what is a factor ma? Factor always occurs in pairs with clear contrasting characters. Gene is also occurring in pairs, and that pair is called as alleles. Gene is a combination of alleles. Each allele is coding a trait. Each allele is coding a trait. So alleles can be dominant and recessive. Dominant allele always masks the effect of recessive allele. Homozygous and heterozygous. Homozygous alleles will have either pure lengths, capital T or small t. Heterozygous means it is a combination of alleles. In the combination of alleles, always dominant allele will mask the effect of recessive allele. Now looking into the phenotype and genotype. Phenotype is nothing but the morphological appearance of any organism is said to be phenotype. Genotype is nothing but the genetic complement that we give to any organism or to multiple characters. Genetic complement which we give to multiple character is called as genotype. We will understand phenotypic ratio, genotypic ratio much better when we are dealing with monohybrid, dihybrid and trihybrid crosses. Clear ma? Now this is the meaning of all these terms. I hope this is very much clear to you. Now to explore the dimensions of inheritance and variation in a scientific method, to explore the scientific method of inheritance and variation, Gregor John Mendel has conducted hybridization experiments, on pea plants from the year 1856 to 1863, which is almost 7 years. And after conducting monohybrid and dihybrid crosses, he has conducted monohybrid, dihybrid and trihybrid also. But now we are learning, in this chapter we will learn monohybrid, and dihybrid crosses. From monohybrid cross he has given or he has derived two laws of Mendelian principles. One is law of dominance and the other is law of segregation. From dihybrid cross he has derived one law, that is law of independent assortment. So yes, this is very much important. Make a note of it ma. What are the two laws that Mendel has derived from monohybrid cross? Law of dominance and law of segregation. What is that law which he has derived from dihybrid cross? That is law of independent assortment. Clear? So this you keep it in your mind. So why did Mendel has taken pea plant for his hybridization experiment? Why did he choose pea plant for his hybridization experiments? He has chosen pea plant. It is because, why did he choose pea plant? He has chosen pea plant because it is an annual plant. It is an annual plant where it completes its lifespan, in just 1 year. It completes its lifespan in just 1 year, so that the results are easily visible, so that the results are easily calculated. And the second most important feature is they show clear contrasting characters, be it dwarf, or tall, be it about flower color or pod shape, seed shape, anything it might be, clear contrasting characters are visible from a pea plant. Okay, and third thing is they are bisexual flowers. They are bisexual flowers. In pea plant, it's a bisexual plant, bisexual or monoecious. Bisexual or monoecious, where it conducts self-pollination so that pure lines or true breeding lines can be seen, and we can easily conduct cross-pollination or artificial cross-pollination can easily be conducted. And when Mendel has observed all the successive generations in his hybridization experiments, he has clearly seen the stable trait of inheritance in all the successive generations of the hybridization experiments. These are the 5 major reasons why Mendel has chosen pea plant, that is Pisum sativum. What is the scientific name of pea plant now? Pisum sativum. Make a note of all these characters, important for objective. Clear? Why did Mendel choose pea plant? Okay. Next. And very important chart, students, you can take the screenshot of it or just fix it in your mind. That are 7 characters or traits are assumed. How many characters? 7 characters or traits are seen. What are those 7 characters and traits? Flower color, seed shape, seed color, pod shape, plant height and flower position. These are 7 characters or traits which has 14 variants. How many variants ma? 14 variants. Of these variants, it is very important for you to know which is dominant and which is recessive. Which is dominant and which is recessive. Okay, which is dominant and which is recessive, you have to know it clearly. Let us assume flower color. Flower color, purple is dominant. Which color is dominant ma? Purple is dominant. When we consider seed shape, round is dominant. When we consider seed color, yellow is dominant. Yellow seed color is dominant, which is this. Okay. Pod color, green pod color is dominant. Pod shape, inflattent is dominant. Plant height, always tall. And flower position, if it is arising from the axial bud, not from terminal bud, that axial flower is dominant when we consider flower position. Terminal flower is recessive, which arises from the terminal bud. Okay, and the recessive characters include, for flower color it is white, seed shape it is wrinkled, seed color it is green, pod color it is yellow, pod shape it is constricted, and plant height is short. When we consider the chromosome number, Pisum sativum, a diploid cell of Pisum sativum has 14 chromosomes. How many chromosomes ma? 14 chromosomes. 14 chromosomes. If we consider haploid set of chromosomes, 7 chromosomes, right? Haploid set of chromosomes, 7 chromosomes. So in these 7 chromosomes, seed shape occupies 7th chromosome. Seed shape is present in 7th chromosome. Flower color and seed color are present in 1st chromosome. Pod color in the 5th chromosome. Pod shape, plant height and flower position, these genes are present in 4th chromosome. These characters are exhibited by genes present on 4th chromosome. You have to remember this, fix it in your mind, very much important for calculations also. Not all the time tall, dwarf, round, wrinkled and seed shape is given. Okay. Sometimes flower position, with respect to flower position also question is asked. With respect to flower color also question will be asked. With respect to pod shape also question will be asked. So keep it in your mind, it is not always seed color, seed shape, plant height. Any of this can be asked for your calculations. So you have to understand, you have to fix it in your mind, all the dominant and recessive traits beta. Next. How did Mendel conduct the hybridization experiments? How did he conduct the hybridization experiments? We have already discussed, no? Pea plant has what type of flowers? Unisexual or bisexual? Bisexual flower. Bisexual flower means it has both androecium and gynoecium. And rhizome has filament and anther, no? Before the anther releases pollen grain, or before microsporangium in the pollen grains get microsporangium in the anther gets matured to the pollen grain, this anthers are dissected, anthers are removed or dissected. This process is said to be emasculation. This process is said to be what beta? Emasculation. Once they are emasculated, these are said to be emasculated flowers, and they are bagged properly. They are bagged properly, so that the pollen from other flowers will not reach out to the stigma of the emasculated flower. Clear ma? Now, when we are emasculating or we are removing the anthers from a dominant trait, here the dominant trait is flower color, that is purple, right? That is purple. Now, we will clearly choose the pollen of a recessive trait. White is the color of a flower. White is the color of a flower. From this white flower, we will take the pollen grains and we will dust this pollen grains on the emasculated flower. And when we collect the seeds of them and sow those seeds in the first generation, the first plants whichever we get from this cross pollination is said to be F1 generation or first filial generation. All the F1 generation plants which Mendel observed are showing only one character or only depicting one parent. They are resembling only one parent, that is dominant trait. So, F1 generation is showing only one of the parental traits, that is dominant trait is what he has observed. Okay. So, what we are doing ma? First we are emasculating the anthers, second we are bagging the flower, third we are dusting the pollen of recessive trait on the stigma of this emasculated flower. Third, we can conduct either monohybrid, dihybrid or trihybrid crosses. How we can conduct dihybrid and trihybrid crosses? For the flower which we are emasculating, we will choose any two traits. Let's assume flower color is already there. Let's assume seed shape, round seed shape. So, the plant which has purple color and round seeds, that plant we will choose, and we will dissect the anthers of that plant. And one more plant which is white and has wrinkled seeds. Which seeds ma? Wrinkled seeds. From this pollen we will take and dust it over here. Here it is 2 gene inheritance, and then we will conduct the hybridization experiment. And third, lastly we will collect the seeds and analyze the phenotype and genotype of particular generations. Clear ma? How did he conduct the experiments? Next. So, yes, monohybrid cross just now we have learnt. So, one thing you should be clear about is in parental generation always parental generation is always homozygous, dominant and recessive, because they conduct self pollination. So, because of the sequential self pollinations, pure lines are obtained. The homozygous tall is crossed with homozygous dwarf. Okay. So, yes, in F1 generation we will get heterozygous tall. That is why all the plants in the F1 generation resemble only one of the parent, that is dominant parent. Okay. And when we conduct the self pollination of F1 generation, then we get okay, self pollination of F1 generation, capital T, capital T, small t, capital T, small t. Okay. So, what are the combinations that we are getting in the F2 generation? One is homozygous tall, two are heterozygous tall, and one is homozygous recessive. This is what ma? Genotypic ratio. And for phenotypic morphological appearance, if you see, 3 are tall and 1 is dwarf, that is 3:1. What is that? 3:1 is what ma? Phenotypic ratio. Clear beta. Next, if you see, it is Reginald C. Punnett who gave us the graphical representation. It is who ma? Reginald Reginald C. Punnett gave us graphical representation to clearly depict the possible unions of all gametes, all the possible unions of gametes, and thereby we can easily we can easily take down the phenotypic and genotypic ratios of every cross. Okay, this is called as Punnett square, which is discovered by Reginald C. Punnett, where we can analyze all the possible unions of gametes and we can take down the phenotypic and genotypic ratio. So, if you take the F1 generation, in F1 generation, of course true parental lines are pure breeding lines, that is capital T homozygous dominant, homozygous recessive. Then in F1 generation we got all heterozygous dominant traits, all the heterozygous dominant genes wherein phenotypically it is resembling only one of the parent. In F2 generation plants resemble both the parents that is tall and dwarf. If you look into the phenotypic ratio that is 3:1 and genotypic ratio 1:2:1. Just now we have observed that. So, yes, from the monohybrid cross, what is the phenotypic ratio, what is the genotypic ratio is definitely asked, and in your question not all the times tall and dwarf is given, any of that 7 traits, any of the 7 characters are taken and the questions can be asked. So, you have to remember all the 7 traits and characters. So, yes, from this monohybrid cross, Mendel has given us 2 laws. Mendel has given us how many laws 2 laws. One is law of dominance and the other is law of segregation. Law of dominance always says that factor occurs in pairs, which shows clear contrasting characters. If the pairs are heterozygous, if the pair is heterozygous, one will be dominant and the other will be recessive. The dominant allele will always mask the expression of recessive allele. And the second point is in F1 generation, only one parental character or one parental trait is resembled or observed. Okay, and in F2 generation both parental features are observed, both parental traits are observed. Okay, and here in F1 generation or in F2 generation, whenever there is a contrasting pair, he said dominant allele will affect or it will close the effect of recessive allele. Dominant allele always mask the effect of recessive allele, clear? And this point is not always valid. So, law of dominance is not universally accepted. It is not universally accepted. If you take law of segregation says that whenever 2 gametes are crossed, okay, the traits are never mixed or blended. There is no blending of characters, Characters separate, okay, all the alleles or traits get separated during the time of gamete formation and each trait is expressing their own feature, but there is no blending of characters or traits, okay, and traits separate during gamete formation, and there is no mixing or blending of characters or traits. This is what law of segregation. Law of segregation is universally accepted. There is no exception for this. There is no exception for this law of segregation. Clear? Now you got to know what is law of dominance and law of segregation. In law of dominance, what he is saying, each factor occurs in pairs. If this pair is clear contrasting traits, if this pair is coding for clear contrasting traits, dominant allele will mask the effect of recessive allele. And in F1 generation, only one parental trait is expressed. In F2 generation, both the parental traits are observed. This is what we are discussing in law of dominance. In law of segregation, what they are saying is, at the time of crossing or combination of two traits, there is no blending or mixing of characters, okay, and traits separate during gamete formation. Traits separate during gamete formation, and each trait is individually expressed. This is universal, this is not universally accepted. Important point to be noted. Clear ma? Next, if you see, if we want to know, sometimes if we want to know the genotype or phenotype of F1 generation, if we want to know the genotype or phenotype of F1 generation. In F1 generation, let us assume all the dominant traits are seen, only one parental generation is expressed. In F2 generation, both are expressed. So in F1 generation, whether to know all offsprings in F1 generation, offsprings resemble only one parent, right? So this parent, when they are resembling only one parent, whether this is homozygous dominant or heterozygous dominant can be observed by this test cross, can be identified by the test cross. What is identified ma? In F1 generation, whether it is homozygous dominant or heterozygous dominant is observed in F1 generation. Let us assume in F1 generation we got homozygous dominant traits. This in a test cross means F1 generation is crossed with a recessive trait, or heterozygous dominant we got, it is crossed with a recessive parent. What is a test cross? Crossing any organism or any gene of F1 generation with a recessive parent, is called as test cross. Crossing of F1 generation or any organism with the recessive parent is called as test cross. What is the benefit of test cross? We will get to know what is the genotypic ratio and phenotypic ratio. What is the genotype particularly of the F1 generation? If it is homozygous dominant, all the offsprings of the test cross will be expressing the dominant trait only. All the offsprings of the test cross will be expressing the dominant traits only. If it is heterozygous dominant, okay, then half of the parents will be dominant trait, and half of the offsprings will express the dominant trait, and half of the offsprings will express the recessive trait, that is 1:1 ratio. Half of the offsprings will express what ma? Dominant trait, and half of the offsprings will express recessive trait. You will see genotypically and phenotypically 1:1 ratio. When you observe 1:1 ratio, then all the offsprings or the tested offspring is having heterozygous dominant feature or dominant genotype. It is heterozygous dominant when you get 1:1 genotypic ratio. When all the offsprings are showing only dominant trait, then it is homozygous dominant. Clear ma? What is a test cross? F1 generation crossing with a recessive parent or any organism crossing with a recessive parent is called as test cross. This is to identify the genotype. Clear? So yes, if the tested organism is heterozygous dominant, you will get 1:1 ratio. If it is homozygous dominant, all the offsprings will resemble only one dominant parent. Clear ma? One dominant trait. So this is about test cross. Next, incomplete dominance. Now we will discuss the deviations of Mendelian laws of inheritance which he has derived from monohybrid cross. One is law of dominance and second is law of segregation. Of course, from those two laws, law of segregation is unanimous, there is no exception for that. But for law of dominance, there are exceptions which we are going to discuss now. one is incomplete dominance. Let us assume the flower color ma. Flower color. What examples we are using to understand each phenomena is important. Keep examples in your mind. Flower color of snapdragon or Mirabilis jalapa. These are two different plants which show this incomplete dominance. Mirabilis jalapa, which is also called as 4 o'clock plant. Okay. So, these two plants flower color, if you assume, the dominant color is red and the recessive color is white. When they are crossed, you know, when their pure lines are crossed, the F1 generation we got is pink. The F1 generation we got is what ma? Pink. According to Mendelian laws of dominance, whenever there are contrasting traits, dominant allele should mask the effect of recessive allele, but here that is not happening. At the same time, mixing or blending of the characters is also not happening. What is happening over here? The dominant allele is partially expressed. That is the reason we call it as what ma? Incomplete dominance, not complete dominance. Okay, where the dominant allele is partially expressed, not completely expressed. That is why in F1 generation we will see pink flowers. When all these pink flowers are self-pollinated, we got one red flower, two pink flowers and one recessive, that is white flower. So, here in incomplete dominance, the genotypic and phenotypic ratio, are same, that is 1:2:1. One is red, two are pink and one is white. So, here in a flower, if you see snapdragon or Mirabilis jalapa, here you can observe all the red flowers, all the pink flowers and all the white flowers too. Okay, all the red, pink and white flowers you can see clearly. So, this is the result of what ma? Incomplete dominance. So, incomplete dominance is a phenomena which is deviating the Mendelian's principle. Okay, and the second concept which we are going to learn, this is also a deviation from Mendelian law of inheritance, that is codominance, where two alleles are equally dominant, or this can be explained by ABO blood grouping, blood grouping. So, if you take type A blood group, A-type blood group will have A antigen, and antibodies are anti-B. Okay, B blood group has B antigen and anti-A antibodies. AB has AB antigens and no antibodies. O blood group has no antigens but anti-A and anti-B antibodies. Okay, so now when let us assume, so ABA blood group has three alleles ma, IA, IB and IO. Okay, IO can also be written as I only. So, one character or trait is expressed by many alleles, not just two alleles, many alleles. So, this is also an example for multiple allelism. This is also an example for what beta? Multiple allelism. What is multiple allelism? Multiple allelism means to express one character, there are more than two alleles, so it is a concept of multiple allelism. How codominance can be explained, you see now. Let us assume male parent and female parent combinations of blood group. Male parent has IA, IB or IA, IB and IO. Female has IO blood grouping or IB, next IA, IO and IO. Let us assume one more, that is IA and IB. These are possible, all the possible combinations which I have taken for both male and female parents. If you look into the zygote, or the offspring, zygote or offspring, IAIO, okay, and here the combination is IBIB. Here again the combination is IA. Here again the combination is IBIO. Here again the combination is IO and IO. Here again the combination is IA and IB. Okay ma. So here if you see clearly, A and B, when A and B blood group is present, it is equally expressed. When A and B blood group is present, when A and B alleles are present, IA and IE, when these two alleles are present, this is equally expressed as IAB, IA and IB, which is coding for AB blood group. This is coding for what blood group ma? AB blood group. So here it is showing the phenomena of codominance, okay, where two alleles are equally dominant and two alleles are equally expressing, for example, AB blood group, okay, and multiple allelism can be explained as this one character is coded by more than two alleles. Now we will also see on how to calculate the genotype and phenotype for multiple allelism. To calculate the phenotype for multiple allelism, it is just n plus 1. Formula is n plus 1. To calculate the genotype for multiple allelism, the formula is n by 2 into n plus 1. So if you clearly see, again if we take male and female parents, and here it is offspring, IAIO, IAIA, IBIO, IBIB, okay. So what are the combinations that we are going to get? IO is one combination, IA is one combination, IO is one combination, IB is one combination, IB is one, and IO blood group is one combination. So here it is what blood group? A. Here also it is A blood group, here it is B blood group, B, and here it is AB, and here it is O blood group. So if you look into the phenotype, let n be the number of alleles, What are the number of alleles coding for ABO blood grouping? What is the number of alleles? IA, IB and IO. 3 alleles, right? So 3 plus 1 it is 4. 3 plus 1 it is 4 phenotypes. Yes, it is 4 phenotypes, A, B, AB and O. 4 phenotypes. If you take genotypes 3 by 2 into 3 plus 1 means 3 by 2 into 4. 4 3s are 12 by 2 is nothing but 6. There are 6 genotypes. Of course, there are 6 genotypes. 1, 2, 3, 4, 5, 6. 6 genotypes are present. And normally to analyze, types of alleles, not for multiple allelism, types of gametes, to know about types of gametes, the formula we use is 2 to the power of n, where n is number of heterozygotes, heterozygous alleles. Clear? So all these formulae, keep it in your mind. We are going to exercise in the numericals in the coming session. So clear beta, you have understood incomplete dominance where 2 alleles are partially expressed. Co-dominance, that is in IA, IB where two alleles are equally expressed, that is co-dominant. Third is multiple allelism where a single character is coded by many alleles, that is again ABO blood grouping. We can use this as the same example, okay. And the last concept which is a deviation from Mendelian law is pleiotropy. What is pleiotropy? Pleiotropy means one gene, one gene is expressing multiple traits. One gene is expressing multiple traits. Multiple traits or characters. Multiple traits or characters is called as pleiotropy. Let us assume starch synthesis, wherein capital B, it is synthesizing large starch grains and the shape is round. Capital B, small b, it is also intermediate size of the grain and it is round. Small b, small starch grain which is wrinkled, So if you clearly look into it, capital B, small b, it is showing intermediate starch grain. Starch grain is intermediate but round. So this intermediate starch grain means again what ma? Capital B is partially expressed. So here particularly it is showing incomplete dominance. Starch synthesis or starch grain, if we consider as a character, then capital B, small b, capital B, small b, all are expressing multiple traits. Multiple traits. In homozygous dominant, it is large and round. Heterozygous dominant, intermediate and round. Homozygous recessive, it is small and wrinkled. So as there are multiple traits expressed for one gene, this is an example of pleiotropy. And one very good example of pleiotropy is autosomal recessive disease. Autosomal recessive disease, that is phenylketonuria. This is because of absence of one enzyme, that is phenylalanine hydroxylase, wherein the aromatic amino acid, that is phenylalanine, this phenylalanine hydroxylase will convert this phenylalanine to tyrosine. It will convert the phenylalanine to tyrosine, so that the functional properties of the amino acid are expressed properly. But then because of the absence of this one enzyme, phenylalanine gets stored in the form of phenylpyruvic acid. It gets stored in the form of phenylpyruvic acid in the brain cells. Because of that, mental retardation. Second is frequent micturition. Third, reduction of skin pigmentation. Skin pigmentation is seen. Okay, what is observed ma? Because of the one enzyme, means one gene is coding for one enzyme. Just because of the absence of one enzyme, multiple characters are shown. One is mental retardation, frequent micturition and skin depigmentation, okay, reduction of skin pigmentation. As multiple effects are shown because of the absence of one enzyme or one gene, this is also a very good example of pleiotropy. And in Drosophila also you can say white eye mutation, only because of the absence of one gene. It can also show multiple effects, like its body size will be differed, its body color will be differed, okay. So yes, this is one good example, and starch synthesis stands the example for both pleiotropy, and intermediate starch synthesis is also an example of incomplete dominance. So if you clearly look into the deviations of Mendelian law of dominance, first it is incomplete dominance, second it is codominance. Incomplete dominance, the example we have taken is what ma? Flower color of snapdragon or Mirabilis jalapa. Codominance and multiple allelism, the example we have taken is ABO blood grouping. And to prove pleiotropy, we have taken two examples. One is starch synthesis and the other is phenylketonuria disease, which is autosomal recessive disease. In coming class, we will discuss more clearly the tricks and the concept about dihybrid cross and the deviations from law of independent assortment also. Thank you.